So maybe we could start and then we're going to record and people will
Yes,
your presentation.
no problem, as you
Yeah.
prefer.
So talk a little bit about yourself, your lab, what you do.
Yes,
Yeah,
okay.
we have been friends for like what 10 years
10
already.
years, yes, 10
Yeah.
years. Yes, yes.
Yeah.
Well, hi everyone. Marjorie, hello. Well, my name is Maximiliano Giraud-Billoud. I work in Mendoza at the EM Institute of uh, CONICET and University of Cuyo. Here,
Oh,
I will.
Tanya is coming. Oh, that's good.
Hello, Tanya. And well, I work essentially with with a mollusk that is a invasive species in all of the world, and it's very interesting because it have many mechanisms to tolerate different kind of stressors. So I will talk today about estivation, but in another occasion maybe I could talk about another experimental models that we have here. Um, Thank you very much for being here today, and thanks for the club, on Redox Biology, for inviting me to speak at this seminar. It is real pleasure for me to share with you some of our research, and this talk is titled Adaptive Physiological Responses to Hypometabolic Conditions Induced by Estivation in an Invasive Organism. First, we can say that animals, regardless of their habitats, are exposed to different types of environmental stressors. These often constitute selective pressures that determine the biological niche of certain species, as well as their ability to occupy new niches. Frequently, the most common environmental stressor for animals in their habitat are seasonal changes, leading to periodic adaptations of varying duration. In some cases, animal species are exposed to extreme living conditions throughout their lives, leading to the development of physiological adaptations over long periods. In this image here, we can see some llamas from the northern Argentina, from Jujuy, and these animals inhabit high altitude all their lives. So they're very interesting, and as these are very interesting animal models in the biology. It is also important to recognize that the anthropogenic impact of human activity on the environment causes changes that expose animals living in suitable biological niches to stress conditions of different durations. In some cases, these changes can lead to the extinction of a species in their native habitats or necessitate adaptive mechanisms to overcome them. Many of these stressors, regardless of their origin, induce a period of quiescence in animals during which hypometabolism is associated with more or less prolonged periods of hypoxia, leading to modification in animals' redox metabolism. The key point is that both the hypoxic state during quiescence and the transitions out of it can increase the production of free radicals. Therefore, the adaptive response of each organism to this redox imbalance determines the toleration of this species to the different kind of stressors. So we could divide or we could say that the animals are divided in hypoxia sensitive animal species and hypoxia tolerant animal species. In hypoxia sensitive animal species, we have the production of oxyradicals that depends of the changes between normoxia and hypoxia conditions. In this paper, very interesting paper from Eaton and Parmenter, they postulate two kinds of conditions related to hypoxia-sensitive animals. They say that in A, we can have an experimental model, an animal, that is exposed to increase in ROS production during hypoxia, and this hypoxia it's maintained during a chronic period. In B, is postulated that the ROS production is predicted to increase during the pass from normoxia to hypoxia, and also with the pass from hypoxia to normoxia, and the animals have another increase of another overproduction of ROS during reoxygenation that also is deleterious for them. This is some examples from these hypoxia-sensitive animals. For animals that are adapted to hypoxia states, these animals can survive a lack of oxygen for hours, days, weeks, or even months, and the production of ROS in hypoxia tolerant is not so deleterious from this animal species than the others. So the authors, Eaton and Parmenter, again postulate some kind of different states from these animals. In A, we can see that species are adapted to permanent hypoxia, like I tell you with the example of the llamas, and are very kind of examples in biology. In B, we can have an animal species that is adapted to hypoxic seasons, so they show a metabolic suppression with decrease in ROS production, but when the animals turn from hypoxia to normoxia again, they have an increase of ROS to the baseline. So the difference between B and C could be that under hypoxia and the change from this state to normoxia, the animal experiments a new increase in ROS overproduction. And the last example proposed by these authors is that the animals, similar to hypoxia intolerant species, suffer an increase in ROS when the animal changed from normoxia to hypoxia, and again from hypoxia to normoxia. But the increase in ROS during reoxygenation in hypoxia tolerant species are expected to be reduced compared to those in similarly exposed in hypoxia-sensitive animals. So, as oxygen concentrations declines from normoxia to anoxia, cellular hypoxia occurs, and at some point during the hypoxic phase, the ROS formation temporarily increased. During reoxygenation, as oxygen concentration rise from anoxia to normoxia, ROS formation also increase. At both moments, the increase of ROS are expected to cause oxidative damage and activate antioxidant defenses. Those ROS are likely the signaling molecule involved in this preparation for oxidative stress, and it's very interesting that this strategy has been carried out under controlled laboratory conditions, and almost all the studies have used a specific time of stress. And in 2022, more than 100 species have evidences of the use of POS strategy to cope with oxidative stress. Multiple studies have demonstrated an elevation of endogenous antioxidant defenses under conditions such as estivation, hypoxia, hibernation, and this response involves species of vertebrates and invertebrates that are distributed across 9 different phyla. So new studies are focused on describing this, that are the mechanism behind this response. For the last 20 years, part of our research group has been dedicated to studying the physiological adaptation of a highly invasive species of mollusk. The Institute EM is located in Mendoza, Argentina, in our province, in addition to Mount Aconcagua, that is the highest mountain in America. We have beautiful landscapes, and consider that people call our province the land of the sun and the good wine. So you should consider to visit us. Well, back to science. Our experimental model is Pomacia canaliculata. This animal is an ampullarid snail, and this species has a native distribution in tropical and subtropical regions, with a distribution mainly in Argentina, Bolivia, Paraguay, Uruguay, and Brazil, but it has invaded different territories and has achieved widespread global distribution, like we can see here. This snail is the only freshwater gastropod among the 100 worst invaded agents in the world. It has the ability to colonize, survive, and invade different habitats. It has separate sexes. Here we can see a female and a male. And they have a high reproductive rate with aerial oviposition. Each female could lay 200 to 400 eggs in each oviposition, and every female can lay eggs 2 or 3 times a week. So in certain times of the year. So it's very impressive, and it has a negative effect in each place that they colonize. They have a lifespan around 2 or 4 years, and they have a biological and morphological characteristics that favor survival. They are amphibious and polyphagous. In our institute, we have a colony of this species of snail that is originating from Buenos Aires. We also have a biotherium for reproduction and growth. And we have an experimental one that is used to make all of assays that we made in the lab. But it is important that this snail can be easily sampled from natural populations due to its size and because it inhabits the shallow water of lakes, rivers, ponds, and swamps. Here we can see some pictures from lakes from Mendoza and the animals in their natural environments. This freshwater snail, given the environments it inhabits, may be exposed to different environmental stressors such as estivation, hibernation, salinity, pollutants, predator, etc. And as this species is amphibious and can survive out of water because it has both a lung and a gill, the first stressor we modeled in our lab was estivation. Is keeping the animals out of water for different periods. So we observed that when the animals spend longer periods without water, their mortality increased exponentially. And after 6 weeks, the animals shows damage or evidence from damage occurred by oxidative stress. So we decided to explore this kind of model in our experimental animal, first in 45 days of estivation. Our experimental model is similar to the animals that are out of water, and well, here you can see an animal estivating outside the water in natural condition, and here is in a vessel in our lab. So initially, our aim was to determine if tissue oxidative stress due to an increase in free radicals occurs during hypometabolism and hypoxia of estivation, or if it happens during the reactivation of the animals in the arousal. Additionally, we investigated antioxidant defenses and other protective factor like heat shock proteins. Under a long-term estivation or estivation from 45 days, we observed that after this period, the animal did not use antioxidant defenses, enzymatic antioxidant defenses like superoxide dismutase or catalase. These animals employ essentially non-enzymatic antioxidants to tolerate the oxidative stress, with uric acid playing a significant role. This species has a specialized tissue characterized by our team in the past, which remains or which maintains a constant exchange between soluble uric acid in hemolymph and intracellular deposits of uric acid. So this is important to and continuous exchange and the availability of the uric acid as antioxidants. Also, we studied the participation of heat shock proteins in the protection of the proteome during hypometabolism, and we observed that both HSP of 70 kilodaltons and 90 kilodaltons increases their levels after 45 days of estivation. We later wondered if the lack of activity of enzymatic antioxidant during hypometabolism could be associated with the temporality of the response to oxidative stress, as it was logically to think that the degree of stress and hypoxia or ionoxia was not the same in animals exposed to short or long periods of metabolic depression. So we conduct a laboratory test exposing animals to only a week of estivation, and after 7 days of estivation, we observed that total free radicals increased in hemolymph, and there was evident damage in lipids and proteins due to the increase of oxidative oxy radicals. Also, the activity of antioxidant enzymes during this short period of estivation increased their levels in the tissues studied, and the non-enzymatic antioxidant didn't show significant changes. So this indicates for us that by drastically reducing the stress time, the antioxidant response differed. Also, with this short mode of estivation and knowing that this animal employs the strategy of preparation for oxidative stress, we found it interesting the study of the possible molecular mechanism involved in this response. To do this, we measured by real-time PCR the expression of some redox-sensitive genes such as FOXO, HIF, and NRF2, and we found that during estivation, FOXO was the only gene with an increased expression when we compare animal under estivation versus control. While the other two showed no significant changes or a diminished like, and a decrease in the expression like we found in the gene. Summarizing the results of the redox metabolism characterization assays in the estivation of Pomacia canaliculata, we can say that the antioxidant defense response varies depending on the duration of the hypometabolism. In short periods, where antioxidant enzymes are associated with protecting against the excess of free radicals, the increase of these defenses can be linked to the increase in the expression of FOXO3. While in 45 days of estivation, the protection is made by essentially an increase in non-enzymatic antioxidant defenses, and the mechanism leading to this response is still unknown. We suppose that could be related with activity of enzymes like xanthine oxidoreductase or xanthine dehydrogenase, but we're not studied yet. Well, to not boring you too much today, I will stop here. However, before finishing, I want to give you a brief spoiler of a possible another seminar if you are interested, obviously. And because we are studied another stress model, and is related to exposing these species to cold. Given the wide global distribution of this species that is exposed to a broad range of temperatures, this snail is not tolerate freezing, but it has a significant survival rate above 5 degrees Celsius. So we explore the response to hibernation, and here is some animals in the referee, and here are some animals hibernating naturally in a lake from Mendoza in winter, where the water was at 8 degrees Celsius and the lake was covered by a nice layer approximately of 3 millimeters. So we studied both times of response at 7 and 45 days of hibernation, and we studied the antioxidant defenses, but this could be for another talk. And another interesting and recent results of our lab are related to the response or comparison between animals inside the referee versus the animals under the ice. So these results could be discussed in another meeting, and well, this is not yet published. So thank you very much. I really appreciate your attention and participation.
Oh, thank you, Max. Thank you very much. It's very nice to see these differences between long-term, 45 days, and short-term estivation. When you were starting your presentation, there was a slide that you're showing T-bars in different
Yes.
weeks of estivation. Is there a significant difference in when you compare 1 week with like 2 weeks, 3 weeks?
Yes, we observe a significant difference between 1 week and every week until the last period. Here is we see
Yeah, yes.
we observe
Is this higher than the others?
what?
Significantly higher?
Yes, yes,
One,
yes.
2 weeks,
The
3 weeks. Huh?
asterisks indicate that are significant changes. And after 2 weeks are significant, and 4 weeks are significant, 6 weeks are significant. So we decide the time of estivation related to the mortality of the animals. After 6 weeks, the animal mortality increased a lot.
Yeah, one thing you might be interested to explore are some pharmacological inhibitors of antioxidants. We have been doing this for some time with different species. Aminotriazole that inhibits catalase, so that could give you some interesting results. So what happened to your snail when it is depleted of catalase?
Yes,
Pharmacologically, and
in a
let's
short
say
period,
two
probably.
weeks compared to 4 weeks of estivation, you might get different results. And also you can test the BSO, yeah, inhibitor of the synthesis of glutathione. Uh,
Yes.
yeah, we published a paper on snails
Yes,
last
yes,
year.
yes. I saw it. I see it.
Yeah,
Yes.
we didn't see much stuff in our model. We tested the hypoxia, yeah, severe hypoxia, almost anoxia, and the only difference we find with BSO was an increase in the redox actually in the ratio of oxidized to reduce glutathione, so the animals are in more oxidized state in the cell, but we didn't see difference in actual damage. Uh,
Mhm.
but you might find more interesting stuff in your models, because we were working with tardigrades. So when you inhibit catalase in tardigrades, it has a devastating effect in these animals.
Mhm.
Because tardigrades, they go into a severe metabolic depression, and then they wake up later. You
Mhm.
and if you add this pharmacological inhibitor, so they still go into dehydration, but when they wake up from dehydration, they just die.
So
So yeah, yeah.
they didn't
So
tolerate the stress.
yeah, so we had to reduce very much the dosage, so we able to see actual biochemical differences, because what we see is measure. It's a genocide. We
Yeah.
have a tardigrade genocide.
Yes.
So you might get interesting results with your snail. Yeah. So
Mhm.
it depends when animal is too tough, you don't get much results. That's the snails we work here, but you might get you might get luckier.
Yes, it's very interesting idea.
So please I open for questions.
Yes, I hear you.
So I think people is a little bit shy. Yeah.
It's okay.
Okay,
But
Basil.
congratulations for your results. We'll stay in the same slide. And I would like to ask you
Yes.
how you can explain this sharp increase in T bars within the first 2 weeks, and whether there is any correlation with the metabolic rate.
Yes. I think that
I mean, I mean because we know that the metabolic depression takes time, isn't like turn off.
yes.
So
Mhm.
you say that during the first stages enter estivation, and the increased metabolic rate proceeded, and then it's going down.
I think that the metabolic depression takes in this species take a long period, and the animal is it going to tolerate in different changes with different tools. I think that the animal first fight with the oxidative stress with antioxidant, enzymatic antioxidants, and then when the enzymatic antioxidant is it's I don't know it's over it's over the non-antioxidants and non-enzymatic antioxidants it's take the control of the situation, and when this last defense is over, the animal it's died basically. I think that I have an hypothetical. I think this is our proposed in 2015 paper related to post responses, and I think that in this species when the animal enters to hypoxia, first we have two increases of ROS, one earlier and other in the reoxygenation. And the first response to this increase of ROS is an enzymatic response. After that, when enzymatic response are over and decrease, probably the non-antioxidant enzymatics check the control and try to make the protection of the tissues and cells from oxyradicals. This is an idea, no? An hypothetical idea.
I don't know the biology of this species, but still this species can respire when it's exposed to air.
I'm sorry, again, please?
This species can
Yes.
respire when it's exposed to air.
Yes. They have a lung and a gill. So, as example, females put their eggs out of water. So there are sometimes out of water.
We should take into consideration the respiration mode of this species, even when it's under start the stage of let's say stressful conditions. Probably there is a first primary stage during which the species, individuals start to prepare to defend the next stages.
Mm-hmm. Yes, it could be possible. Yes.
Yeah. And
Yes.
another question is, okay, we agree with non-antioxidant as non-enzymatic antioxidant, but what are these mechanisms? We know.
I don't know how this snail use uric acid as antioxidant. In other animals and the uric acid acts as a scavenger from oxyradicals and exchange to allantoin when it's oxidized. So we measured in previous assays both uric acid and allantoin, and we found the correlation between them. But the molecular mechanism, I don't know how it's working.
And,
I don't know.
You're right, and I think that probably you should determine some metabolites, osmolytes, you know, which really are characterized as non-enzymatic antioxidants. Last 10 years, several works published about osmolytes and their antioxidant physiological role. And I think uric acid is one of them.
Yes, we need to measure another antioxidants in this species, but are not characterized. And probably we measure glutathione, but probably they have another non-enzymatic antioxidant like ascorbic acid and others that are important in this kind of changes and protections.
Several antioxidant metabolites.
Yes.
And the accumulation of uric acid indicates that you have activation of purine metabolism during long term of estivation, which makes sense about this.
Yes. Probably the synthesis of uric acid is related with the protein metabolism or purine metabolism. So probably if the animal increase their metabolic rate, probably the uric acid also change.
Okay.
Thanks, Basil.
Thanks, Marcelo. That's okay.
Okay, thank you, Basil. Thank you for so So many questions and commentaries. So let's open for other people. So Israel Vega, you could talk to us, yeah?
Yes. We say that cool uric acid could be more related to water availability since it is molecule from nitrogen catabolism and less toxic than ammonium and urea. Yes, probably the animal when tried to keep water and diminish the loss of it, probably uric acid could be the final metabolic pathway from the destruction or the consume of purines, because to uh, excrete ammonium and urea, the animal needs to eliminate uh, some excreta. So to conserve our water, it's probably a good tool, make the crystal of uric acid in the tissues. So Juan Manuel said that Max, thank you. Okay. Bye bye, Juan Manuel. Thank you for assist.
Okay, okay, people, more questions. No, Max is here to answer you. Please don't be shy. Professor Debas, do you have any question? Marco. Nobody has questions. So I think people are kind of shy. So if there is no more questions, we will stop for now and wait for the new data from Max.
Yes, or another,
Yeah,
if you
yeah.
want to talk.
So it's nice. Yeah, just we have been working together for almost, not almost, it's 10 years at
Yes,
least.
10 years.
Yeah,
Yes.
a long time. Yeah. So it's nice to see how this is going. It's very nice. Yeah. Uh, so 10 years ago, there's a lot of mixture. We didn't know things. We are just
Yes.
postulating mechanisms. So now some of these ideas are coming true. Yeah, we're
Yes.
seeing results. Yeah, and
And
our hypotheses are being confirmed. By,
yes.
I mean, generally, yeah.
Yes.
And
We're
so,
working a lot.
are you planning to do any work in nature following our latest paper on
Yes, in hibernation, but is not published yet. This is in nature. I'm working with animals from here, Mendoza, and I'm comparing the both results and hibernation in the lab versus hibernation in the nature. So this probably will be published when we finish the determination.
so
But
you
it's very
finish
interesting.
and did you get differences? Just curious.
No, not spoiler.
Oh, no,
Here,
no, no spoiler.
no spoiler, not spoiler.
Okay, no spoiler.
It is very interesting the molecular response. That is it the only spoiler that I will tell you. We studied the antioxidant, the activity, and also the expression of redox genes. And in genes, we have some kind of differences with estivation. So maybe the stressor is linked to different kind of mechanisms also. So we're we're trying to understand the mechanism right now. Okay, I don't know if you don't have any more questions. We lost Marcelo, but
Okay, I'm back.
okay.
Okay,
Hi,
sorry.
Marcelo. Yes, it's okay.
So, do we have more questions? I think we are okay. So thank
Max,
you. Oh,
Max, sorry.
Yes, Marcelo.
You start working under the field conditions?
Under what?
Field condition.
I don't understand.
So,
Under natural conditions, yes. This is I talked with Marcelo that here this is under natural conditions of in hibernation. In estivation, we're not working yet because the animals, when estivate, are they put in the land. So we have some troubles to find the correspondence between natural estivation and our lab, because we put out of water. It's very more stressful our condition than in the nature, because when the animals are in the land, they have a mood and some humidity that in the lab they don't have. So in estivation, no, we don't have any natural or field experiments.
Okay. We'll be waiting for your results.
Yeah, okay, thanks.
okay. So thank you, Max. Thank you, everybody, for coming. So we will soon release the video for your friends to attend, watch, and we will see for another seminar very soon. Yeah.
Okay. Bye bye.
Okay.
Thank you all.
Bye bye.
Bye bye.
Thank you, everybody. Bye bye.